Hello everyone, I am Vicki Kretzschmar with the Auburn Huntsville Research Center, and on behalf of the Auburn University Research Advisory Board, and more specifically the Research and Industrial Relations Committee, we are pleased to welcome you to our webinar today, and we appreciate your attendance. And for those of you who may not know, the Research Advisory Board is composed of more than 40 industry professionals from across the country who volunteer their time very generously to support the research mission of Auburn University. And they do that in many ways. A lot of them is just going out and identifying ways that companies can utilize our researchers and the specific things that are being developed at Auburn University. And they also work within the university with administrators to find creative and innovative ways to support and incentivize our research faculty and students. And that mission part of the Research Advisory Board is very much the reason why people like Dr. Lipke are with us at Auburn University, and we're fortunate enough to have her with us at Auburn University so we can attract those types of faculty to Auburn. And so we'll get started with a couple of housekeeping agendas. The webinar today will last for approximately 1 hour from 2 to 3 o'clock. All webinar participants are initially on mute. You'll see that on the sidebar of your screen. If you would like to unmute yourself, you would just have to click on that and you can ask questions if you would like to do that. We'll have a question and answer session at the end of the presentation, so I ask that you hold your questions until that time. But if you don't have voice capability on the webinar, you can submit your question in the question section, and I will moderate and answer those and ask Dr. Lipke for you. The recorded webinar may be viewed afterwards on the Auburn University Research Advisory Board website, which can be found at the link listed below. And there's also a history of the webinars that we've had so far. We've had 8 of them featuring faculty from across the university, anywhere from nutrition to materials. So if you have interest in that, you are welcome to go back and check out the historical archives of the webinars. You're encouraged to pass this webinar link to your colleagues and contacts and anyone that may have interest in Dr. Lipke's subject matter. And if you know people or organizations that may have interest in the research topic, please pass that information along to Dr. Lipke at her email address there. You may also find her on her website throughout on the College of Engineering. And also, if you would like to contact me directly, I'll be happy to point you in the right direction. At the end of the webinar, there's going to be a short survey which will be emailed to you following the webinar, and we ask that you just fill that out for us and help us streamline this a little bit better and respond to specific needs that you may have or topics that you may have that interest you for upcoming webinars. Our next webinar is tentatively scheduled for May 14th, and that's all depending on the status of graduation and where we can identify faculty that are not taking a vacation that week. And right now I'd like to introduce our speaker today, who is Dr. Elizabeth Lipke. We're very fortunate to have her at Auburn. She is an assistant professor in the Department of Chemical Engineering at Auburn University. She did her graduate studies at Rice University under Dr. Jennifer West in the Department of Chemical Engineering. Her research was in the area of biomaterials and cardiovascular drug delivery. And after completing her PhD, Dr. Lipke was a postdoctoral fellow at Johns Hopkins University in the Department of Biomedical Engineering, where she created and investigated the electrophysiological properties of cardiac sheets created using stem cell-derived cardiomyocytes. Dr. Lipke has been an assistant professor in the Department of Chemical Engineering at Auburn for approximately 5 years. And during that time, her research group has been recognized for their work, winning many awards, including Dr. Lipke, who recently received an NSF Career Award. Today, Dr. Lipke's topic will be addressing engineering tissues to understand and treat disease. I'll turn it over to you, Dr. Lipke.
Thank you. just make sure that you can see my slide.
Yeah, here they come. Okay.
Great. I think you should be able to see my slides now. Is that the case?
Yes.
Great. So thank you very much for the opportunity to talk to you today about some of our ongoing research and how that fits into the field of engineering tissues to understand and treat disease. I hope you'll come away from this perhaps understanding a little bit better what the challenges and also the opportunities are by creating tissues outside the body, and then also to understand, of course, how researchers at Auburn are contributing to this field. And so I put a picture of myself on my first slide because I've never given a webinar before. I think it's a little strange to not know how many people might be looking at their cell phone at any given time. So I will do my best to keep this interesting and at the right level for a broader audience. So if you would like more detailed information, I'll just put that at the beginning about any of the research that I talk about. I'd be happy to provide that, but I haven't tried not to burden you with unnecessary amounts of bar graphs and schematics today. So when we think about understanding and treating disease and using engineered tissues to do this, I have here two of my students, Shantanu Pradhan and Alex Hodge, and they're both working on creating engineered tissues. Shantanu, can you see my can you see my pointer or not?
Yes.
The mouse or just?
I can see both.
Okay, great. Shantanu is working on creating engineered tumors for a wide variety of applications, and Alex is working on creating engineered heart tissues. And so I think that with their two projects is a good illustration of the answer to why engineer a tissue. And so if we think about classical tissue engineering, in the classical sense of tissue engineering, and I know I don't look all that old, but I feel like I've been in this field forever since I started as a freshman. And so it's a developing field that's been around for maybe 20 years. And you think about the fact that we would first remove cells from the person's body. And in classical tissue engineering, the idea was that we would expand these cells outside the body, provide them some materials to grow on, and then after providing the cells some materials to grow on and perhaps some stimuli to help the tissue form, we would re-implant the tissues back into the patient's body. And so when I think about this, I kind of think about it as the freshman experience at Auburn, right? If you think about tissue engineering, it's kind of like taking a freshman from their parents' house. So inside your body, your cells are getting very specific cues. They're getting signals, they're getting nutrients, they're getting oxygen, potentially. And that's like the freshman at home. The freshman has rules. Before they come to Auburn, they have all sorts of checks and balances from their parents. And then the parents drop them off in their first freshman dorm. And that's kind of like this step of taking the tissue, the cells out of the body. And so, of course, the parents, they really want the child to continue following the rules at home, doing the things that they used to do, just like we would like the cells potentially to follow the rules that they were following inside the body. And some of the things freshmen do all by themselves. Most freshmen continue to take a shower. Most freshmen potentially do laundry, assuming that the parents provided them with the detergent and so on and so forth. And that kind of goes back to the same thing for the cells. Some of these cues that we are giving the cells are receiving inside of the body, they don't necessarily have to have us create for them. They know those things, and they'll continue to do those things over and over again, even after we take them and put them inside the dish. But some of the things that the cells we're doing inside the body are going to require that we provide stimuli and information to the cells so that they will continue to function the way they did inside the body. If you have 5 roommates, you might start studying at the library as opposed to studying in your room. If you live on the 5th floor dorm and the elevator is broken for 5 months, you might look very different than if you live on the 1st floor and are eating at Chick-fil-A every single day. And the same thing can be said for the cells. So as tissue engineers, one of the things that we talk about is how we can control the environment that the cells are provided in order to help them replicate their function, their structure, and their behavior that they would have in their native environment. So there's two reasons that we would like to do this, and those reasons have actually changed a bit over time, and I think they have a varying impact. So first and foremost, I think we can all understand that we would like to repair damaged or diseased organs inside the human body. We've been talking about this as a field for a long time. In particular, as I mentioned, Alex Hodge in my group is interested in regenerating heart tissue, as well as one of my other students, Patrick Kircher. But this is quite challenging in terms of the amount of tissue, as you'll see, that we need to prepare, and all of the other things that happen after we've implanted the tissue. So just to give you the motivation, of course, for repairing the heart, we have, second, my slide doesn't want to advance, probably because it thinks I'm trying to write something. There. Okay, so most of you probably realize that contemporary treatments, of course, do not regenerate the heart. Heart disease continues to be the #1 cause of death for both men and women in Western society, and very sadly, the number of young people with heart disease has gone up. Oh, sorry about that. All of a sudden, I think there's a little bit of lag and when I tell the slide to go forward and when it actually does. Sorry. So the number of young people with heart disease is increasing rapidly, with even young children before their teenage years experiencing some of the detrimental effects of our Western culture. So the issues that contribute to heart disease: coronary artery disease, heart attack, which is basically causing death of your heart tissue, cardiac arrhythmias, congenital heart defects, and congestive heart failure, really are treated symptomatically. They aren't repaired for the most part. And so tissue engineering has the potential to help us in understanding the repair of these things. The other reason to engineer tissues is for the creation of model tissues. Sorry. For the creation of model tissues for advancing biomedical research. And in this case, we would like to create tissues outside of the body that are similar to those inside of the body. And this is being done both for cardiac tissue, so heart tissue in my lab, and for cancer tissue. And so when we think about tumors, they have very complex structures. They have a multitude of different cell types. They have blood vessels. They have supporting cell types. And all of these factors come together to regulate how the tumor cells are going to respond to various treatments. And so by creating engineered tumor models outside the body, we can perhaps create models that are better replicative of what's going on inside the body, and therefore better able to predict which pharmaceutical treatments might be effective, either overall or even on a patient-specific basis. And so if you think about tumor cell culture outside the body, on the left you see the cells growing. This is a breast cancer cell line growing in 2D. And on the right you just see them in aggregates. And even these aggregated cells are more similar to a tumor structure than of course that 2D flat cell that you see on the left. So it's kind of like expecting you to walk on your hands and knees on the floor all the time and have the same physiology as you would if you lived your life in 3 dimensions. Of course, we all live our life in 3 dimensions, and so it's very helpful to replicate these 3-dimensional environments, and we can use materials to even further replicate the environment that the cancer cells experience inside of the body. So the next question is really how we create engineered tissues. This is Patrick Kretzschmar, one of my students, and she's working on engineered heart tissue as well, particularly human heart tissue. And so she's sitting by the microscope looking at the computer, and you can see her little tissues here back on the scope. When we create engineered tissues, we usually are combining both the materials, whether it's a natural material or a synthetic material, the cells, and some signaling molecules or bioactive factors. We then have our tissue engineered construct. We can treat it in a way that's stimulating pathological environments, for example. And of course, we would then like to probe this engineered tissue for a number of things. We could use it for drug testing and toxicology testing. We can use it to understand human development, which is particularly difficult to get at in the body, of course. We can use it to understand mechanisms of disease, and we can use it to come up with new therapeutic interventions. And I'd like to just point out that in terms of therapeutic interventions, this is both for our veterinary patients as well as our human patients, that therapeutic interventions through tissue regeneration and regenerative medicine are of high interest. So if we just take our first component, the cells, right, they have, they're pretty essential whether they come from the body itself or whether we grow them outside the body, and take a closer look at the cells. We can see that if we have adult cells, their regenerative potential is limited. So if cells have a high turnover rate, they have high regenerative potential. And if they have a low turnover rate, they have relatively low regenerative potential. So in our lab, one of the high turnover cells that we're working with is endothelial progenitor cells. And we also work on a collaborative project with Dr. Wooldridge in the College of Veterinary Medicine looking at equine endothelial progenitor cells for regenerative medicine and regenerative applications. And so those cells can actually be isolated from blood and expanded and then reused for investigation and eventually clinical treatments. However, when we look at our other applications, heart, these cells have very low cell turnover. Cardiomyocytes, the contracting cells in your heart, there's some really exciting news recently. 0.01% of the time they're dividing during childhood. So if you can imagine, most of the cells that you have in your heart today that contract, the ones that do the pumping, and we're chemical engineers, we all like our favorite pump, which is the heart. Most of the cells that are doing that pumping don't divide within your lifetime. And so if you have a heart attack, you lose a large number of those cells. Your body has several hundred billion cells. Your average organ has 100 billion cells to 1,500 billion cells. And so in a heart attack, you might lose 7 billion cells. So this is clearly an issue of scale-up and an issue that's important. And how are we going to get all of these contracting heart cells if they don't divide? So one of the challenges for tissue engineering is cell source. And of course, for cancer cells, they are very happy about dividing. So those are particularly for our heart tissue engineering, we need to have another cell source that is not an adult cell type. So we've turned to stem cells. Just as a review, a stem cell is a cell that can self-renew and has the potential to become at least two different things. So some adult stem cells have the potential to become bones and fat, for example, or new blood vessels. In particular, so we need to use something called pluripotent stem cells to create those contracting heart cells, the cardiomyocytes. And so we use these pluripotent stem cells that come from a biopsy, for example, of human or animal skin. They're then genetically reprogrammed. And at that point, we can then train them using our biomimetic materials and bioactive factors to become a range of cell types within the body. In fact, because they're pluripotent, almost every type of cell within the body. So they have a huge potential for use in cardiac and other types of regenerative medicine where adult stem cells are more limited. they kind of, if you need to have an analogy, you might have cast, I kind of like the use of analogy. An induced pluripotent stem cell is like taking a PhD chemical engineer, and of course highly specialized, like our cardiomyocytes, the contracting heart cells. And basically we send those cells back to kindergarten. And that's where we get to hear, take our PhD chemical engineer and we send them back to kindergarten through the reprogramming step. And as those kindergartners then, that's the point at which we work with them in our lab, as those kindergartners are then trained, they can become a wide range of things. And so we would like to have a concert oboist, for example. And so we're really interested in how we can give the cells the right cues for training to become the concert oboist. And unfortunately in the field, in most cases, really what we get is a third grader. And so one of the major challenges in tissue engineering and really going from these two-dimensional training protocols, which we call differentiation, to a three-dimensional tissue engineered system is that we would like to enhance the maturity of the cells so that they're more similar to the cells that are in the adult body. So if you need an analogy for induced pluripotent stem cells, it's just the sending of cells back to kindergarten. So, and when we take our tissue engineered scenario or tissue engineering, for example, of the heart cell, and back to that classic idea of regenerating tissue, in this case, we call the myocardium, the muscle of the heart. We take our cells, and if we're tissue engineers, along with some materials and other factors that give the cell signals, and then these things are injected through this little bitty needle and incorporated into the wall of the heart. And the idea is then that they're going to contribute, of course, to the pumping action of the heart and increase cardiac output. Up until now, most clinical applications and clinical trials, of course, have looked at their cells. And so the challenge with that is that most of the cells don't actually make it. They die at the point of injection, or they don't remain in the place of injection. And this is really true for tissue engineering applications or regenerative medicine applications, both in the human realm and in the veterinary realm, and not just for the heart, but all tissue types. And so maybe 10% of these cells actually are still living and in the location you want them at the end of the day, 24 hours or so post-injection, depending on the tissue type in the study. So I would like to make this analogy to the space shuttle, basically sending the cells into the body without the help of any supporting factors or materials. Is kind of like sending the astronauts into space without the space shuttle. It's just very hard on the cells, and it's, of course, from the cellular perspective, not the astronaut perspective perhaps, it's really hard to keep the cells where you want when you get them there. Body is an extreme environment, just like space. So, in our lab, we're using biomimetic materials to control the environment around the cells and to provide them with those cues that they would be receiving if they were still inside the body. And some of these cues include bioactive factors, degradation sites, cell adhesion sites, mechanical stretch, electrical stimulation, and even changes in topography. And we've been able to show that all of these things are able to influence the cell in terms of its function and tissue formation. So basically the take-home message is that materials can provide cues to direct the differentiation of cells, so their specification processes, and how tissues form. And so the goals of our research, and the research that I think I failed to mention, but this is an extremely collaborative type of research, of course. There's engineers, there's biologists, there's clinicians, and then the many different types of engineers that come into the picture, material scientists. And so the goals of this research are really to drive the maturation of the cells, the formation of tissues that replicate native tissues to reduce the heterogeneity between cells over time and to facilitate the scale-up of production and eventually, of course, to provide a platform for supporting the cells in their next location. And so our lab has done a number of things. We've worked on delivering nitric oxide, where I'll tell you a little bit about today. We've worked on nanopatterning, and I'll show you one of those examples. I haven't included an example of our electrical stimulation studies. We have some of that work published, and we also work ongoing looking at conductive polymers, if you have interest in that. I'm going to start by telling you about our hydrogel microsphere system and our systems that we are using for cell encapsulation. And we gear all of this to be able to put it eventually in these scalable surge flask bioreactors so that we could go from the millions of cells that are typically created in flat plates to the billions of cells that would be needed to actually create engineered tissues for drug testing applications or actual repair of some part. So the base material that we work with in our lab is polyethylene glycol. Many of you are probably familiar with this. It's advantageous for tissue engineering because it's biocompatible, it's non-thrombogenic. Probably most important to us is that we can mix our cells with it and then cross-link it using visible light or UV light at physiological temperature and pH. So it's kind of like making Jello in that you can mix your cells with it, and then like the Jello, the cells are distributed throughout. So I'm originally from Minnesota, and you kind of have that picture of the Lutheran potluck, right, with the fruit salad Jello as a staple ingredient. Of course, like Jello, our polyethylene glycol hydrogels have a high water content. This is really important for the cells because it facilitates the nutrient and the waste exchange. And important for our research is that we can modify and change out the peptide ligands, the drug delivery ligands, and all the different portions of the hydrogel backbone quite readily to study the effects of different components. We're primarily working in the projects I'm going to tell you about today with PEG-fibrinogen, which provides the cell adhesive sites and degradation sites and is pegylated so that we can cross-link it relatively quickly. So one of the things that we use for culturing our cells is microspheres, and this is basically an aqueous PEG-fibrinogen precursor solution that we then use to make a water and oil single emulsion. We have our surfactant in our oil, and then photoinitiators, one in the aqueous phase, one in the oil phase. We gently vortex for 2 seconds, and then shine a light on our polymer solution to create our spheroids with encapsulated cells. And so we've shown that in this research that we can vary the number of cells encapsulated in the spheres by varying the cell concentration. So these here are these water and oil created hydrogel spheres, and the small dots are the cells within the spheroids. And as you can see, as we increase the initial starting concentration of cells, we get more cells in the spheroids. And so this is important just to create, for a number of reasons, that we wanted to create an environment that could be used in those scalable culture systems for differentiating the cells, as well as an environment that other groups have shown these spheroids are injectable for therapeutic use. So they go through the needle. We were able to show then these are mouse pluripotent stem cells, that the cells grow within these spheroids, and then what was important to us was contract. Oh, sorry, the spheroid is supposed to be contracting. I'm not quite sure why it doesn't contract today. It always contracts, something happened, and contract inside the spheroids. So you can see when we immunostain these, they have beautiful cardiac markers, as well as Connexin 43, which is a gap junction. that is necessary for cell-cell connectivity, and they show striations of alpha-carbonic actinin. So the cells go from being these pluripotent stem cells to functional cardiomyocytes. And so in the next figure, I do have, these are human pluripotent stem cells contracting, that differentiated into cardiomyocytes, the contracting heart cells, so cells that were sent back to kindergarten from human skin cells, and then in our lab differentiated, so directed to become these contracting heart cells. This is a cell island here on the left, as we call it, and here's a larger scale, as you can see from the scale bar, a larger scale tissue that's contracting in the lab after the cells went through the process of being specified into these contracting heart cells called cardiomyocytes. So the material, of course, is one of the factors that direct the differentiation of tissue formation. A couple of other factors that we have looked at for directing tissue formation along with collaborators include bioactive factors and electrical stimulation, as well as surface topography. And so I'm going to show you an example of the delivery of nitric oxide to the cardiomyocytes. So we delivered nitric oxide to our differentiating cardiomyocytes. Nitric oxide has to be delivered locally because it has a very short half-life. It's very important for a number of native cell signaling mechanisms, so it's important in your brain, in dilation of your blood vessels. It's important for platelet function. It's important in your immune system, in your kidneys, and reproductive system. And it also is important in directing stem cells into becoming specialized cells, in this case cardiomyocytes. And so this work is actually in preparation, so I'm not, and actually was recently submitted. So we're not going to go into it in much depth because I'm sorry I can't really publicize all the figures until they're out there. But basically what we've done is taken a suspension culture of our differentiating stem cells, provided them with this nitric oxide donor in a way that's very similar to the not-scalable flask system. Because the nitric oxide can diffuse readily through the cells, it's a really great small molecule. We can also couple it, of course, to the backbone of the polymer, and we were able to get an increased yield of stem cell-derived cardiomyocytes, including increased numbers of contracting spheroids, this typical way of culturing the cells, an increased frequency of contraction of these spheroids. And from the electrophysiological side, or the calcium transient side, we had decreased calcium transient durations, indicating that they have better calcium handling than controls. So it's just an example of how a bioactive factor could direct these differentiating stem cells. Another important feature of cardiac tissue is its multi-scale organization, and for that matter, any kind of tissue. So here we have just a schematic from one of our recent review articles looking at how actin-myosin filaments then build to myofibrils, to these contracting myocytes, all the way up to your heart, or if you start with the heart, you can go back down. So from the macro scale to the microscale and the nanoscale. And so when we see at our cardiomyocytes on nanopatterned polyethylene glycol hydrogels, here we have on the left the unpatterned hydrogels, and on the right the patterned hydrogels. You can see that the cells in fact align along the patterns. And when you look at conduction, when you look at conduction and action potential propagation through these tissues, you will see that there is also an effect on the electrical propagation through the tissues. So, and as I said, we also have electrical stimulation data that I haven't included today. So when we go back to all of the different aspects of our engineered tissue that we'd like to control, one of the things we're interested in is drug testing and toxicology and therapeutic interventions. And so this really relates back to Shantanu's project in the tumor cell and tumor engineering project. So as many of you probably understand, chemotherapeutics are highly toxic both to cancer cells, fortunately, and to cardiac tissues, unfortunately. And we would like to create these engineered tissues both to understand how to make or create platforms so other people can understand how to make chemotherapeutics more effective, and also to understand how we can prevent some of those toxicities by using these engineered tissues. And so one of the things that we've done in our lab is take what we learned from the cardiac project and translate it to our tumor culture system. And so what we created is an encapsulation system for encapsulating tumor cells in these three-dimensional hydrogels. You can see them here. This is right after the tumor cells were encapsulated. These are breast cancer cells, but we've done this now for actually quite a range of different types of tumor cells. And you can see the cells grow inside of these what we call tumor microspheres. And over time, eventually, they start to grow out of the tumor microspheres by day 28. This time course of cell growth in our 3D tumor microspheres is much longer than you could grow these cells in a two-dimensional setting if you want to do drug testing in a dish. So that facilitates a much longer time frame for looking at the effects of drugs on the cells. And so some of our collaborative projects are interested in that aspect of things, as well as it increases and improves the microenvironmental conditions that each cell is experiencing to make those conditions more similar to what you would find in a native tumor tissue. So the cells at the center of these tumor microspheres are experiencing hypoxic conditions. So you can see here is a tumor microsphere after 28 days of culture. Here in green, you can see the live cells. In red, you can see the dead cells. And so this is that classic formation of an area where the cells inside of the tumor are dying. With that, we created these cells, of course, using that initial aqueous in oil emulsion system that I showed you at the earlier in the talk. These tumor microspheres, of course, don't have such a great large area of cell death as you might expect in a native tumor. Their size ranges from 100 microns to 500 microns, but still they have more similar cues than to cues the tumors that are formed by self-aggregation of cancer cells, and also not all types of cancer cells self-aggregate. So one of the advantages of the system is that we can incorporate multiple different types of cells. We can incorporate cells that could provide vasculature, for example, or support cell types in addition to the tumor cells. Fibroblasts, for example, could be incorporated into these spheroids, and we're also interested, of course, in how the mechanical properties play a role in the phenotype of the resulting cells. In order to increase the size of that hypoxic core, my student Shantanu came up with a new fabrication method for creating what he has coined as hydrogel millibeads. These millibeads give us a much wider range in terms of the size of the microspheres, and in this case, millispheres. So one of the disadvantages of the previous system was that we couldn't get the large sizes that we needed, and so he showed basically by this system where he dispenses his hydrogel onto the surface of an oil bath, and then shining his fluorescent light onto the surface, onto the millibeads, that he can photocross-link them and really tightly control the size of the beads that are produced, as well as get these larger size beads that were not originally possible. So here we have a picture of the uncrosslinked beads floating on the surface of the oil. Here are the crosslinked beads, and here are our 10 microliter, 5 microliter, and 2 microliter beads photo cross-linked next to a dime for scale. And so we've also shown that we can incorporate cancer cells into these millibeads and then culture them, again, in a dish, varying, of course, the concentration of the cells and the size of the millibeads. And here we have some SEM images. Here is an SEM image of one of the tumor millibeads. Here's an up-close image where you can see the cells on the surface, as well as, if you look closely, some of the remaining material. And what was important in this project is that we now have this larger area of red cells, which are the dead cells, creating the larger hypoxic core that's on more of a size scale that we're interested in mimicking. So this is several millimeters in diameter. So this is another example, of course, of these then could be used for drug testing in toxicology, or testing, even in the case of personalized medicine, for therapeutic interventions. The PEG-fibrinogen that we use as the base material for these has been shown by our collaborator to have potential for culture of primary tumor cells from patients, and we're looking into that in the longer term. As well, of course, as understanding mechanisms of disease. And so when I get to my final slide, you'll see we have a long list of collaborators that have been interested in using these structures, as well as a provisional patent on the fabrication method for studying cancer tumor engineering. Okay, so one of the important aspects, of course, to both cardiac tissue regeneration and to cancer is understanding blood vessel formation. And so the final thing that we're working on in our lab and in our tissue engineering project is vascularization and understanding blood vessel formation. And so one of the things that we're doing to understand blood vessel formation is working with the endothelial progenitor cells. These are the lining cells of your blood vessel. Basically, they're the coating of your blood vessels, and they're also responsible for making new blood vessels. And so what we're doing in our lab is understanding how these circulating endothelial progenitor cells can be expanded or just captured under blood flow and used for tissue engineering applications. So in one of our projects, we look at rolling these cells across the surface of our materials, which can be, of course, coated onto the surface of the blood vessel. This has been shown by previous research in other groups, and then modifying our materials to incorporate specific peptides that interact with specific ligands on the surface of these cells in order to understand the behavior of how they roll, how they adhere, and how they eventually capture onto these surfaces. And so we do this using a parallel plate flow chamber, where we have our peptide grafted hydrogel on the bottom. Here's the parallel plate flow chamber at the top, where we flow our solution, mimicking blood flow across the surface of the material, and then we flow our cells through this parallel plate flow chamber and examine how the cells adhere to the bottom material. And so I'm going to show you an example of the cells rolling across different peptide coupled surfaces. So we have 3 different peptide coupled surfaces that we've investigated, REDV, RGDS, and YIGSR. These are typical peptides for culture of endothelial cells in the literature, and they all interact with different integrins, or in the case of YIGSR, a non-integrin receptor on the surface of the cells. And what we found was that by varying the type of peptide on the surface, we see first of all, that these peptides support the rolling of these cells that are isolated from blood, and second of all, that the type of receptor that they're interacting with, or the exact peptide that they're interacting with, affects the rate at which the cells roll, with the slowest rolling being on this red V-coated surface, and then the other two peptides supporting cell rolling at approximately the same velocity. What's really unique about this system is because we start with the polyethylene glycol base, we inhibit cell protein absorption, and so we can really look specifically at these peptides and their interactions specifically with these cells. And so we don't have to sort of subtract out all the absorbed protein on the surface of our PEG, because we've done a lot of control experiments to show that that's not a factor in the cell rolling process for these endothelial progenitor cells, specifically endothelial colony forming cells. And so we've also worked, and I haven't shown the data today primarily because it's unpublished and it's collaborative with Dr. M. Wooldridge in veterinary regenerative medicine, to isolate these cells from horses, these endothelial colony forming cells, along with our student Maggie Salter. And we've shown that these cells, of course, are able then to act as endothelial cells, that they are endothelial cells, and that they can be used for vascularization right now, just in our 3D constructs, which is nice, but eventually our next step, of course, is to look at whether they're going to help with revascularization, where revascularization is a key contributor to tissue regeneration. As I said, particularly bone formation requires revascularization, as well as a number of other difficult to treat diseases in humans and in horses. So I would like to conclude by saying that tissue engineering holds promise, not only as a means to repair damaged and diseased organs, but also as a platform to study development, human development in particular, that's hard to get to in other ways, and test pharmaceuticals. And cell material interactions are playing an important role in just guiding this process of tissue formation and function. With that, I'd really just like to acknowledge all of the people that are in my lab, particularly my graduate students and undergraduates, as well as point out some of the Auburn University collaborators that are helping with us with this work. So I mentioned Dr. Ann Wooldridge on our equine regenerative medicine project. She's an expert in that field. Dr. Zuming Zhang, who's helped us with the calcium transients. Dr. Chris Easy, who's also a collaborator on the cardiac project for some of the human engineered cardiac tissue that I haven't presented. And then for the cancer tissue engineering, Dr. Alan David, who's collaborating with us to use our tissue engineered constructs for testing his particles. Dr. Michael Green, we're working with him, as you've heard in a previous lecture, to create some tissue engineered models with his lab. Dr. Rusty Arnold and Dr. Mark Byrne, also in cancer collaborations, and Dr. Zong and Dr. Hamilton in our conducting polymers work with the cardiac tissue engineering. And of course, I'm very appreciative of the funding from NSF and the American Heart Association. And please feel free to contact me. Here's our web address, as well as you have my email address. I'd be happy to take any questions.
Wonderful. Thank you, Dr. Lipke. That was just fascinating work that's going on in your lab. Again, let me remind you, if you have a question that you'd like to submit, if you don't have voice capability, you can type it on the right-hand side in the question panel, or you can unmute yourself right next to your name with the green microphone button. Does anyone have any questions? Sometimes it takes a few minutes for people to find where the green button is. We'll let them look.
That's pretty fast. They're off.
All right, at this time I would like to open up the floor to Stan Graves, who is a member of the Research Advisory Board and also serves as the chair of the Research and Industrial Relations Committee. Mr. Graves? Are you with us?
Yeah, thank you very much, Vicki and Dr. Lipke. It's always fascinating to find out the exciting things that are going on in the world of Auburn research. Can you share with us kind of the level of funding you have on these two projects from NIH and also the American Heart Association, and are there other venues for funding that we might be able to help you with?
So we actually have funding from NSF and the American Heart Association. Funding from NIH is definitely the goal of my goal, the goal for this type of research. And we do have a pending proposal with an SBIR where we are helping, basically, of course, but more opportunities like that to really provide our expertise to go to industry, to create these three-dimensional tissues that I think the trend is going towards in terms of both industry as well as research funding would be beneficial.
Have any of the biotech firms showed an interest in this at this early stage?
So we have an ongoing proposal with computational fluid dynamics in Huntsville.
Okay.
Is that who the SBIR?
Yeah.
Okay.
So that would be, we're providing the three-dimensional, basically the three-dimensional sides of the engineered tissue constructs. I mean, one of the advantages, particularly, for example, of the cancer project, one of the big challenges in creating engineered tissues is that the cells, one of the challenges from the 2D perspective is the cells don't have very long to grow. One of the challenges from the 3D perspective is that you can't control the size if you just do self-aggregated spheroids. And so causing some problems. And so by being able to control the size really tightly and make it very uniform, it makes it more translatable in terms of getting a very similar outcome every single time. If your engineered tissues vary from each time you make them, that makes it really hard to use them, for example, for drug testing. And one of the things we found with our human cardiac platform, for example, is that our technique, which I haven't presented, mostly because it's extremely competitive research area and something that we'll hopefully have in review in just a few weeks, but not something that we have published right yet, is that it's extremely translatable. The geometry that we use is we've tried, Petra's tried a number of geometries, and it works for all of those geometries, from the cells that she associates out of the tissues for single cell work all the way up to the macroscale tissues that I showed you contracting. So she can culture the cells in spheroids and get cardiomyocytes. She can culture the cells on little printable islands. For example, all of these tissues are printable for high throughput applications. A lot of them we're creating on acrylated glass slides, so they could be printed onto acrylated glass slides and photocross-linked onto the slides and created that way for high throughput testing. Other people in the field use them in multi-well systems for drug testing, and so it would be very, I guess, rewarding to see some of those applications take root for some of the things that we've developed in our lab.
And I gather from your comments about not disclosing data that you are near filing a disclosure patent on some of the work you've done?
Actually, we have, but it's just even that, so both for the engineered cardiac tissue, the human engineered cardiac tissue, and for the tumor engineering millibead project, we have provisional patents.
Okay.
But particularly in human cardiac tissue engineering, it's a very competitive field, so I just didn't want to put the data slides up there.
Gotcha.
I'm happy to talk about it in more depth. I just
Yeah, well, we don't want to do anything that would create any problems for our competitiveness. We have enough problems as it is.
Well, I guess I felt like we've done all of these things with the mouse pluripotent stem cells as well, and you can kind of see from there where the human engineered tissues would go.
Vicki, I think you indicated earlier we had planned to have 31 attendees. That's wonderful.
We actually had 56.
Wow, fantastic.
Yeah. That's great.
Everybody for participating. As always, I'd like to ask any of the participants if there's any way we can help Dr. Lipke or Auburn University Research. Please contact Vicki or Rodney or Dr. Mason's office, and then I guess we'll see everybody in mid-May in beautiful Auburn.
Absolutely. All right, thanks everyone for attending today, and please join us and share this information with anyone who you think would be interested. I appreciate your help today, Dr. Lipke, and your information.
Thank you very much for the opportunity.
Thank you. Bye-bye.
Bye-bye.